As I mentioned, we're gonna start in 5 minutes so that people can join in. Okay. Oh, there is a workshop. You can attend it. Hindustan Times graphic editor, science communication. Online, it's happening. Zoom.
Engineering helped me decide, or if you realize that I learned more, I learned a lot of science, but I decided to actually go on
Then close it.
of science writing, and through science writing I went into journalism. So just so yes, so it was in fact I did my engineering, and after that I did print journalism immediately. In a year later, I did my print journalism in Kerala. I'm basically from Kerala, but I grew up in Ahmedabad, Gujarat. So but I did my 1-year postgraduate program in print journalism, in Kerala. Then that's where I was exposed to the world of journalism, and that is one of in fact, that's one of the first lessons that I learned personally. That writing is a very, very, very small subset of journalism. So as a student, I was other than yes, I'm a 90. Like to see myself as a 90s kid. So if you're in a typical 90s kid, you have about two options in life. Generally, you have medicine and engineering. If you are academically good in school. So along with these two things, I was always passionate about English and English literature. I generally had a flair for writing.
Don't spill.
Always. But but it's really later when I combined these two that I decided to go for. I want to be a science related writer because I wanted to capture what I learned done in engineering. But when I did journalism is a different cup of tea. It is not really lit. You might to be a good writer is an asset, is a skill. It is useful. It helps you, convey your thoughts and ideas effectively. But the way you write in newspapers is very, very different from the way you write in general. Even in fact, even the whole structure of how you present a piece of news. How do you organize information in a way that what should be coming first, second, and third follows a very different model from what is
Things I have to do for money. Things I have to do for money. Okay, bye. So we're gonna start our today's session. As I mentioned already, that due to some technical difficulties, I was not able to record last class session. So we're gonna repeat that class, and maybe after half an hour, or maybe on Sunday tomorrow, we can schedule 1 hour class to compensate for those things. Okay. So on our revision, we were studying about the secondary structures of protein. We have studied about alpha helices and beta sheets, right? But before coming to the actual 3D structure, as I mentioned that we were discussing about the beta sheets. So in 1951, Pauling actually came up with the pleated sheet, a new layer configuration of polypeptide chains, right? So for many years, the abstract stays. For many years, it has been assumed that in silk fibroin, stretched hair and muscles, and other proteins with the beta keratin structure, the polypeptide chains are extended to nearly their maximum length, which is about 3.6 angstrom per residue. And during the last decade, it has been assumed also that the chains from lateral hydrogen bonds with adjacent chains which have the opposite orientation, okay, a hydrogen bonded layer of this sort is here. So you can see in beta sheets there are two types of beta sheets, parallel beta sheet and anti-parallel beta sheet. So in anti-parallel beta sheet, you can clearly see the organization of hydrogen bonds, that they are a bit more organized in comparison to the parallel beta sheet, which we will see in the coming slide. So this is one polypeptide chain, this is the N terminal of first polypeptide chain, and this is the N terminal of second polypeptide chain. So they run in the anti-parallel fashion with respect to each other. So if this is first polypeptide chain, its N to C terminal is from top to bottom. This is second polypeptide chain whose N to C terminal orientation is from bottom to top, and the third polypeptide chain again is anti-parallel to the second polypeptide chain. So its N terminal to C terminal is running from top to bottom, right? And this is how they are making the intermolecular hydrogen bonding. If you remember, in the alpha helices and the helical type of secondary structures, the type of hydrogen bonding which is stabilizing the secondary structure is predominantly intramolecular hydrogen bonding. The I plus fourth residue type of bonding pattern is there in the alpha helix, while in beta sheet the hydrogen bonding is between two different polypeptide chains, which is called intermolecular hydrogen bonding. Now if you look at the anti-parallel beta sheet, which is depicted here, you can see that they are clearly intermolecular type of hydrogen bonds, but they are bit different in orientation in comparison to the parallel or in comparison to the anti-parallel beta sheet. So if you look here, the N terminal of first polypeptide chain is on the same side as the N terminal of the second polypeptide chain, meaning that they are not anti-parallel, they are parallel to each other, and then they are forming the stabilizing hydrogen bonds. Before further going to the basics of beta or secondary structure, specifically beta sheets, we'll just see a typical overview of the secondary structures via this brief video. I hope you guys can see my screen. Let me share it again. Just give me 1 minute. Yeah, so I'm sharing my screen with you guys. Just let me know if you can see it. Yeah, so we'll start from here.
Talk about the secondary structures of protein, and in that context we would talk about beta plated sheets. Previously we have talked about alpha helices. If you missed that video, go check that video and the link is in the I button. So protein structure can be divided into different structural levels, starting from primary structure, then secondary structure, tertiary and quaternary structure. And now we are focusing on the secondary structural features. Among the secondary structural features, there could be alpha helices, there could be beta turns, there could be beta pleated sheet. The center stage of this video would deal with beta pleated sheet. Now, beta pleated sheet is a region of peptide chain where the amino acids are linked with each other in a specific orientation in form of hydrogen bonding, and this kind of gives like a sheet-like appearance in a 3D space, and that is why the name is beta pleated sheet. So let's look at the structural features of this. So the NH groups of the backbone actually forms hydrogen bonds with the carbonyl group from the other amino acids. So roughly it looks like this. Now, the biggest difference between alpha helices and beta plated sheet is the distance between adjacent amino acid, which is 1.5 Armstrong in alpha helices, whereas it is 3.5 Armstrong in beta pleated sheet. So in terms of steric hindrance, beta-plated sheet structures experience less steric constraint compared to alpha helices. Now, beta-plated sheet can be organized into two different orientations, parallel and anti-parallel. In this particular orientation, you can understand the beta-plated sheet has all the hydrogen bonds in a particular plane, right? In a planar fashion. And all the amino acid R groups are protruding out from the plane either upwards or downwards. That itself is reducing the steric constraint, right? Now let me tell you, due to these kind of structural organization, tryptophan, tyrosine, phenylalanine, these kind of bulky side chain containing amino acid can be a part of beta-plated sheet. But this is not a stereotype. There are many other structures where beta-plated sheet generally prefers smaller side chain containing amino acid as well. And probably due to the structural feature of beta-plated sheet, these kind of bulky groups can fit. Now, if we look at the beta-plated sheet in a Ramachandran plot, we would see the phi and psi angle would be roughly minus 120 degree and plus 115 degree. Roughly, they fall into this particular region. Both parallel and anti-parallel sheets fall into this region. Now the question is, where do we find the beta-plated sheets? It turns out many protein contains both alpha helices or beta-plated sheet as their secondary structures, but the proportion is different. Some proteins might be enriched in beta-plated sheet, while some others are enriched in alpha helices. Just to give you an example, the silk or fibroin, this particular protein, is very much enriched in beta-plated sheet. Now, the supramolecular association of beta-plated sheet has been implicated in neurodegenerative diseases like Alzheimer's, where it can form aggregation. So, beta-plated sheet, their interaction with each other is very important in context of disease. Now, let me tell you that how structural features.
Yeah. So I hope you guys got the basic difference between alpha helix and beta sheet. We'll see another representative video for this so that it's pretty more clear with you guys. Apart from this, we'll move to the technical things of beta sheet. Just give me 1 minute. I'll share the screen with you. So I hope you guys can see my screen. I have muted because the ad is going on. So the overall, what are the major differences between between helix and sheet? And you should ask me that why I am saying predominantly alpha helix and not helix, right? It can be anything. Helix, we studied about two major different types of helices. First one was alpha, and then gamma also we have discussed very less, but still it indicates that there are different type of helices which exist in the biological system. Still we are talking majorly about alpha helix. Why is that so? Because predominantly in the biological system, depending upon the physiological conditions, the majority of the helical structure which are present in the protein, they are of alpha type. Okay, alpha helix. And again, if you remember, we discussed about the left-handed alpha helix and right-handed alpha helix. In that also, the proportionality is pretty clear that one type of alpha helix is dominated in the biological system. Okay, that is why I'm most of the time whenever I say helix or alpha helix, it majorly indicates towards alpha helix only. Okay.
Myoglobin. This protein, as you see, it is made up of 8 alpha helices. In the case of beta-plated sheet, this is silk fibroin that is predominantly made up of beta sheets that is joined by loops. Majority of proteins contain both alpha helices and beta sheets. So this is amylase. As you see, these are the beta sheets and these are the alpha helices that is present in amylase. Now let us see what are secondary structures before moving into the difference of alpha helix and beta sheets. A primary structure is a linear chain of amino acid that is joined by peptide bond. So this is the primary structure. This primary structure undergoes regular repeated folding and forming the secondary structure. And this is primarily formed by hydrogen bonding. The helical conformation is called the alpha helix and an extended conformation is called the beta pleated sheet. These beta sheets are made up of beta strands and alpha helix is the helical conformation of a polypeptide chain. Now let us see the difference between alpha helix and beta sheets. Alpha helix, it is a spiral configuration of polypeptide chain that is formed by hydrogen bonds. As you see, these are the hydrogen bonds involved. Right-handed helices are more common, whereas beta sheets, two adjacent polypeptide strands. These are called as beta strands that are bonded by hydrogen bond. So these two or more strands may interact together to form an extended sheet-like structure called the beta sheet. Now let us see the bonding in alpha helix. In alpha helix, as you see, the bonding is between this carbonyl oxygen and amino hydrogen. This is the bonding. So as a result of this bonding, the structure becomes helical as the amino acids are far away. So this is the second bonding. Carbonyl oxygen and amino nitrogen combines by hydrogen bonding, making the structure helical. Whereas in beta sheets, so this is the first peptide strand and this is the second peptide strand. So hydrogen bonding is between the amino hydrogen of one strand and carbonyl oxygen in the backbone of the second strand or adjacent strand, as you see. So these are some of the bonds. So here also you can see. The third difference is in alpha helix there is intramolecular hydrogen bonding. The bonding is within the polypeptide chain. As you see, all the bonding are present within the polypeptide chain. A single polypeptide chain is involved. Whereas in beta pleated sheet, there is intermolecular hydrogen bonding. There are separate peptide strands or polypeptide chains that forms a fully extended linear sheet-like conformation. So the bonding is intermolecular or between peptide strands. Alpha helix can be a single chain like this. It's a single chain and is of only one type. Whereas beta sheets, there are two types of beta sheets depending on the orientation of the peptide strands involved. So first one is a parallel beta sheets. As you see, this is the N terminal and this is a C terminal. This is the first peptide strand and this is the second peptide strand. If both these peptide strands runs in same direction, this is NC direction, this is amino terminal to carboxy terminal direction. If both runs in same direction, it is called as parallel. Then this is the anti-parallel strand. As you see, this first strand runs in N terminal to C terminal direction and this second strand runs in opposite direction, that is N terminal to C terminal in opposite direction, therefore called as anti-parallel strands. Anti-parallel strands are more stable because of more suitable orientation of hydrogen bonding within these strands. Next difference is regarding the dimensions. An average alpha helix is 10 residues. It can reach between 4 to 40 residues. A complete turn is of 3.6 residues. As you see here, this is the turn and that is made up of approximately 3.6 residues. In beta sheets, a beta strand is a polypeptide chain typically of 3 to 10 amino acids long with a backbone of an extended conformation. Now regarding the amino acids involved. The most preferred amino acids in an alpha helix is alanine, leucine, methionine, etc. And the least preferred is proline, then bulky amino acids like tryptophan, tyrosine, glycine, etc. Proline cannot fit into this helix, very difficult to form the bond and introduces a kink, therefore also called as helix terminator. Proline is often called as helix terminator. In the case of beta sheets, it is an extended conformation, so there is enough space for bulky amino acids like tryptophan, phenylalanine, tyrosine, etc. So these amino acids, bulky amino acids, are commonly present in beta pleated sheets. Hope you understand the difference between alpha and beta helices. If you are benefited, please subscribe this channel and share this video. Take care, stay.
So this was the overview of beta sheet and alpha helices. Now I guess you guys have a little bit of clear understanding like what are the major differences between these two type of secondary structures. There are other secondary structures apart from these two which are predominantly present. Specifically, if we talk about with respect to the membrane proteins, these are the major structures, and among them also helix is more preferred for membranes in comparison to beta sheets, right? But it is not completely true that beta sheets structures are not present in the membrane. Okay, so now you know that we have seen a representation of one structure, right? So it had only alpha helix or a protein has only beta sheet. It is not necessary always. Most of the time the proteins are mixture of these secondary structures, right? So it might have a particular percentage of helix associated with it, beta sheets. So how they are connected with each other? So they are generally connected via each other by small loops. Okay, so before going to that, a very basic properties of beta strand is that in this the polypeptide chain is fully extended. Okay, there are almost 2 residues per turn in comparison to 3.4 or 3.6 residues in alpha helix. Okay, the translation is about 3.4 angstrom per residue. They are stable only when they are in sheet form, right? So a single beta strand cannot exist in the system. Obviously, why? Because if amino acid residues are present in the system, it's a cytosolic system, aqueous environment. So chances are amino acids which do not like water, like hydrophobic residues, they won't be happy there in that kind of structure, right? So when they come in sheets, which means more than 2 polypeptide chains come together and make hydrogen bond for beta sheet structure, they are more stable in that way. Cool. And hydrogen bonds between peptide groups of adjacent strands is formed, indicating that they are intermolecular type of hydrogen bonding and stabilizing the single beta strand in an sheet structure. Okay, and the type, the orientation in which these strands can pair can be parallel or anti-parallel, right? So these are the basic parameters of regular secondary structures. So if we talk about the dihedral angles phi and psi, so in anti-parallel beta sheet, the phi and psi values are respectively minus 139 and 135. For parallel beta sheet, it's minus 119 and 113. Right-handed alpha helix will lie in minus 57 and minus 47 respectively. 310 helix will have a phi psi value of minus 49 and minus 26. Pi helix has minus 57 and minus 70. Polyproline and polyglycine 1, 2, 2. These are different types of polypeptides and whose structures are determined at an angle of minus 83 plus 158, minus 78 plus 149, and lastly minus 80 plus 150 respectively. So you can clearly see there is a huge variety of angles, yet there are few angles which are missing. So this we have discussed in the Ramachandran plot quite briefly, that there are certain regions in the plot which are not allowed. So what do we mean by allowed and disallowed regions? This you know very well. Okay, or we can have a quick recap of Ramachandran plot also. Give me 1 minute. Yeah, I'll just quickly search that thing.
Addicted to procrastination.
Zeal's Pharmacy tutorial, and today we are going to study about Ramachandran plot. I am making this video.
Yeah, this was the one which we saw.
Neil is sitting at a.
Yeah.
They need a way to verify that their structures make sense based on actual allowable molecular geometries. Enter the Ramachandran plot. In 1963, the Indian physicist G.N. Ramachandran described a standard way to define the geometry of a given amino acid in a polypeptide chain. Ramachandran's team noted that some geometries aren't possible due to sterics. In other words, a given amino acid cannot adopt some conformations in a polypeptide chain because of unfavorable crowding with the adjacent residues. And when the two backbone angles are plotted, there are distinct regions for the unique elements of the secondary structure. Phi and psi are examples of dihedral angles, which are, by definition, the angle between two intersecting planes, shown in yellow in this image. Notice that the carbonyl and the adjacent nitrogen are shown with dotted bonds. Remember that peptide bonds, and amide linkages in general, have significant contribution from a resonance structure with double bond character. So much so that the nitrogen and carbonyl group are coplanar. This puts 6 atoms in a single plane on each side of the alpha carbon. Though we don't have free rotation about amide bonds, we can rotate around each bond coming from the alpha carbon atom. Let's head over to our 3D model to explore this. We'll start with a tripeptide made up of 3 alanine residues. And for good measure, let's show the peptide bonds and label the N-terminus and C-terminus. When we measure phi and psi angles for our central alanine, we'll need to look at some atoms in the surrounding amino acids. But we can simplify our structure by removing atoms that are not needed to view phi or psi for the central alanine. Those atoms can obscure other atoms and make the analysis of the angle a little more difficult. The phi angle is defined by rotation about this bond, from the alpha carbon to nitrogen, which I've colored magenta. And the psi angle is given by the bond from the alpha carbon to the carbonyl, which I've colored green. In order to understand the angles, we need to set this up by looking straight at the alpha carbon directly down the bond of interest. So if we're looking for the phi angle, we'll orient the structure like this. And if we're considering the psi angle, we'll have to orient the structure like this. When we look down the bond for the phi angle, this nitrogen is going to be hidden behind the alpha carbon. What we'll be comparing is the angle between these two carbonyl carbon atoms. And when we look down the bond for our psi angle, this carbon atom is going to be hidden. So here we'll be comparing the angle between these two nitrogen atoms. So essentially, we consider a 4-atom pattern when looking at phi and psi. To translate this to...
Yep. So obviously I have already shared this video with you guys. You know what exactly are phi and psi angles. And why it is important to know about the phi and psi angle, why there are only these 2 type of dihedral angles, why they exist, why we are looking at the rotation around the polypeptide or the peptide bond, right? So we know all these things in brief. So as I was talking about that, it is not necessary that the protein will have only helical content or only beta sheet or beta strands. It can have all the structure in all the compositions and combinations you can imagine, correct? So there are loop structures which are not exactly structures, but they are like 3 to 4 residue long chains which just connect one type of secondary structure to the another type of secondary structure. They can also connect similar secondary structures, different secondary structures. It completely depends upon wherever there is no structural content to it, right? So before this, then we have explored the PDB data bank. Again, I'll get back to the main site of PDB. Let me share my screen with you guys. Yeah, I hope you can see it. Yeah, so this is the home page of PDB. PDB stands for Protein Data Bank. As the name suggests, it has all the structures, sequence and related information which till now scientists and researchers across the world they were able to decipher. So there are around 224201 structures in the PDB, and there are around 1068577 computer structure models. So what are computer structure models? These are basically the models which are predicted by a tool called AlphaFold. So AlphaFold is an tool which basically use the substitution or mutation, these studies to predict the structure of a protein which is till now, or whose structure is not there, being submitted to the PDB via experimental procedure, right? So there are few experimental techniques which we have discussed, such as X-ray crystallography, cryo-EM, NMR. These are the techniques through which we determine the secondary structure, determine the structure of a protein, right? So as you can see here, that this is the home page of PDB. There are different tabs which you guys can explore on your own time. There is a molecule of the month, so it's very exciting to see, because in every month there is new molecule, new structure, which are always associated with not necessarily the things which are going on, but I have kept a tab on the molecule of the month panel when we were facing a pandemic called COVID, if you guys remember that, right? So during COVID time, in every month we were having the structures which was here. It was always from which was always related to the SARS-CoV-2 virus. It was interesting to see that how the science is going forward even in such hard times. Okay, so we'll explore one PDB structure, but this is the basic home page. You guys can explore and see, this is a tab here which you can see it says that the recent entries. So even looking at the recent entries tab, you can see how diverse the proteins are. For example, if we see this first one, this predominantly we can see that there are some helices and majorly beta sheet, right? Then next one, so this is the cryo-EM structure. It is completely different from the structure which we saw just in the previous tab, right? You can see how beautiful this structure is and how big the protein is. Okay, again you can see this, which is a virus protein. I assume if you go into the entry of this one, we will know more about this protein. And see, this is such a, it looks like a simple structure, but obviously it's not, because to get even a single structure of a protein, irrespective of whether it's a membrane protein or a cytosolic protein, in itself is a huge task. Okay, so and also if you remember guys, we have discussed about why it is difficult for membrane proteins to be structured, and hence in among these like 224201 structures, there's considerable number of membrane proteins, yet we are still far behind in understanding the membrane proteins from their structural perspective. Okay, if we explore more, see, this one is a crystal structure, so it's a X-ray crystallographic structure. This one is a cryo-EM structure. This is apparently the overall structure of Eastern equine encephalitis virus complex. So you can see how big a protein is, and how from ranging from fewer residue long protein to bulky proteins, everything is there in the PDB itself. And you can see this is such a beautiful protein coming here with a complex with the nuclear protein, I assume. So we'll explore one entry. Why should we not go with the molecule of the month entry itself, right? So we'll just click and see what basic information is present to us here. This is the information about that protein, but we want the structural information. This is the PDB ID. So what PDB ID is? PDB ID is the unique number which is given to each entry, right, which is there present in the in PDB. So let's say you can search via ID, you can search via name. So let's say if we want to see insulin protein, is the most basic protein which all of us have heard about, right? So let's see, and these are some refiners which we can keep, right? So it is saying that structure determination method, we want experimental, because the other type of structural determination will be computer aided, right? So I want to know the Homo sapien insulin structure. I want that only, and let's see what are the entries corresponding to that. So you can see there are different entries. So we'll see the first entry itself, which is 1H59. So 1H59 is the unique PDB ID given to the complex of a protein with IGF-1, right? So the classification says insulin, the organism is Homo sapien, and there are no mutations in it, okay? And this PDB belongs to the corresponding research article in which this structure is reported, okay? So this page will give you the basic information that when the structure was deposited, when it was released, excuse me, who are the authors responsible for this. I'm sorry, yeah. So what method was used for determining this structure? What is the resolution? So resolution basically means that how precisely you can distinguish between two atoms, right? So here the resolution is 2.10 angstrom. The R value free, the R value work, these are some technical terms associated with the way to know that how accurate and how clear the structure is, okay? Then this is the basic representation of the structure. We'll go to the structure tab here. So this is this window basically lets you explore the structure at your will, and how you can move it, rotate it, visualize certain parts of the protein, or highlight some parts, see the sequence, all those stuff. This is a really good visualization tool, but I personally don't prefer it. We prefer separate visualization tool. So maybe in tomorrow's class I'll show you one such protein in that visualization tool, and we'll see how we can use the power of computation to actually understand what's going on with the protein, right? So you can zoom in, you can rotate, you can select residues, you can look into it, what sort of bonds it has. These are the water molecules. We can see that this is a protein which is making hydrogen bonds with the nearby water molecules. We have the sequence here, we have chain information, we have like literally everything which can be of use to you with respect to the structure, okay? And it's a good visualization tool. Then the annotation tab, this is some basic genomics associated information. I'll say that what genes are there, what type of structure it is, superfamily it belongs, all the information, all the related structural information can be found in the annotations toolbar. Then we'll go to the experiment. These are the conditions under which this protein was crystallized, as it's a X-ray diffraction technique used for this protein, and all the information, basic information needed for this, right? Then we have another tab titled sequence. Here you will get the information of sequence, structure, what parts of the structure is being modeled, what part of the structure is not modeled, because chances are if the regions are too small, or the protein is rich in glycine residues, or a lot of hydrogen bonds are there, chances are that you might not be able to see them very clearly in the 3D structure. So even that information is present in this tab. The second last tab says genome. It obviously, as the name suggests, it's gonna tell you about the gene related information with respect to this protein. So you can see structural biology and the structure of protein, or the PDB, is not only the information of proteins itself, it goes to the gene level, and if you want to dig more into it, there are cross references into it. You can go to the gene bank, you can get the genes, you can get the mutations, you can get the structure of those mutations, you can get the proteins which are bounded with an ligand, which are bounded with a drug molecule, what changes are there with respect to that drug, whether when it is bound and when it is unbound. All these information can be very well accessed from the protein data bank. So it's a great repository, and one should actually spend a lot of time in understanding what sort of information you want to extract from this beautiful database, okay? Moving forward, this was a very brief introduction. As you know, we are going through revision classes only, and I'm not discussing in detail about the topics which I'm mentioning here. So this was the data associated with it. It's that this data is till 2022, and you can see how nicely the structure deposited in the PDB are growing, right? Then this we have already explored the information present. So for today's class, I guess I have summarized most of it. I have told you about the secondary structures. I have told you about the information associated with the database itself, and we'll see in the next class, which I'm gonna conduct tomorrow as a compensatory class for 1 hour, obviously. So in that class we might see a simulation of a protein, of a specifically a membrane protein, and how it is working. We'll try to understand some of the structural features from that simulation. And yeah, feel free to ask any question if you have, and you can drop me mail whenever you want. Okay, thank you so much for your attention, and have a good day. Thank you.